has its own characteristic. Am I right? In terms of color, in terms of texture, and many other characters. So we have different faces, different eyes, and so on. So this variation based on variation in the genome, and this variation, and the main source which introduced this variation in the genome is meiosis. So how meiosis create this variation? So assume, okay, every one of us, for example, me, he, and everyone, we are the result of 2 meiosis and 1 fertilization. Am I right? So this is what happened. Okay, so can someone tell me, with these simple, basically, steps or events, 2 meiosis, 1 meiosis which produce basically 1 sperm at the end, actually 4 sperm, but 1 of them is used in fertilization, and 1 meiosis which produce an ovum, and then you get fertilization. How many conditions you expect that such a thing may happen? So the final zygote, which is the result of 1 meiosis in male and 1 female meiosis and then fertilization, how many different scenarios you can expect for this? So the final zygote, how many different forms of this zygote genetically may happen as a result of these 2 events? So you have 1 meiosis in male, and then you have 1 meiosis in female, and then you have 1 fertilization to get the zygote. Am I right? So that's very simple. So you all know that this happened, and you learned this many years ago. But I want to know who thought that how many scenarios you can expect about this. All right, so you.
Is it somewhere around 23 to the power of 2?
Okay, so she says 20 to the power of 2.
23. 23 to the power of 2.
23 to the power of 2. Any idea? Anybody agrees with her? So why you said 23 to the power of 2?
Because you have 52 pairs.
So
Oh yeah, 23.
I'm asking you. So why you said 23 to the power of 2 different scenarios you can expect?
Because you get 23 from the haploid from the mom and the dad, and then that's where the 23 comes from, and then the power of 2 because you get 2 sets, and then the genes, they vary because of different processes. So that's where that logic comes from.
So if you probably think what you said is 2 to the power of 23.
Probably, yeah. Somewhere around that kind of ratio. But it's a big number. It's a large genetic variation.
So, anybody want to add anything to what she said? No? Okay. So we are diploid. Our cells are diploid. It means that a somatic cell in human has 46 chromosomes, which consists of 2 haploid sets. So that's why we call ourselves diploid organisms. Am I right? So what does it mean? It means that for every chromosome, for every autosome, we have 2 copies. For example, we have 2 copies of chromosome 1, and then we have 2 copies of chromosome 2, 2 copies of chromosome 3, and the same, and then we have 2 sex chromosomes, X and Y. And if we look to every of these chromosomes, for example, these are the 2 copies of chromosome 1 in a metaphase, basically, stage, so which we see them like this under the microscope. So every one of us has 2 chromosomes, 1 like this, and one of them is from our dad and one from mom. So, and if you look to the whole these 46 chromosomes, then we have 2 sets. 23 chromosomes from one parent, let's say from mother, so we call them maternal cells. And then we have 23 chromosomes from our father, which we call them paternal cells. Okay, so this is the chromosomal structure that we have in the nucleus of every somatic cell. So, but what happened in the germline? So you know that during meiosis, so these haploid sets, these diploid sets basically get separated, okay? These chromosomes get separated, and finally, after meiosis, what you see in the sperm, in the gamete, is only a haploid set. We have 23 chromosomes, okay? So let's say a gamete is going to take 23 chromosomes from 46 chromosomes that we have here. Okay, for the first chromosome, this gamete has two options. So basically, at the end of the meiosis, the gamete may get this one, get the paternal, or get the maternal copy. So there's two basically options, or two choices for only chromosome 1. So the same happened for chromosome 2. So if you look to the gamete may have this chromosome or this chromosome, because the gamete should have only one of these different chromosomes. If you look to chromosome 3, again the gamete has the paternal copy or the maternal copy. Okay? So for each chromosome, it has two different choices or two different options, two different scenarios. And if you look to the 23 pair like this, the number of scenarios that may happen would be 2 to the power of 23. Am I right?
Yeah.
So this is only for the chromosome. If you look chromosome as a whole, the chromosomal content of the gamete. Okay, so a male gamete has 2 to the power of 23 different, basically, genetic scenarios that may happen to that. Again, if you look to the female gamete, this one also has 2 to the power of 23 different forms. Okay? So 2 together would be 2 to the power of 23 times 2 to the power of 23. So that would be 2 to the power of 46. So this is very simple. That is only just the separation of these chromosomes. But is this the only one which happened in meiosis? So what else happened in meiosis which may increase this genetic shuffling and creating variation, recombination? That is another factor, which is very important. So, and what is this recombination?
The genetic information is transferred, crossing over of genes from one the paternal.
Okay, so it means if you look here what happened, it means that, for example, these are the 2 chromosome 1 that is present in a somatic cell. When a cell goes to be a germline, to be a germinal cell, and enter to the meiosis, as you know, every of these chromosomes make another copy of itself, which is called sister chromatid. All right? So then we have these chromosome 1 with a sister chromatid attached to it. This chromosome 1 also make another sister chromatid. Okay? And then all these 4 chromosomes attach together and produce a structure we call them tetrad. So tetrad formation. So what happened when we have this tetrad formation? Then we get genetic exchange of genetic material between the homolog chromosomes. All right? So you know that these 2 chromosomes that I showed here, that one of them is paternal and one is maternal, we call them homolog chromosomes. Why? Because they are homolog, but they are not exactly the same. Although the overall structure is the same, the loci on these chromosomes for the genes are the same, but in every loci which belong to one specific gene, different form of that specific gene may sit, which called the alleles of the gene. Am I right? So although the overall structure of all chromosome 1 in human are the same, but if you look at which form of the gene sit on each locus, you see they are different. The location of the locus, which is the physical location of the gene, is the same. So what is the meaning of locus? So the physical location that a gene sits on the chromosome. That's called locus. Okay? So for example, if we say that chromosome 1 has, let's say, 5,000 genes, something like that, 2,000 genes. So if you start from one side to the other side, you can see that there are specific seats for each gene. And these specific specific seeds called locus for that gene. So the order of these locus on chromosome 1 is the same for all chromosome 1. Okay? If we get all chromosome 1 that we have here, okay, if we are 60, so we have 120 chromosome 1. And if we look to the structure of chromosome 1, the overall structure is the same. So it means that the order of these loci on the chromosome is exactly the same. If the loci number 10 is for gene A in my chromosome, so the same thing is for his chromosome. It means that the seed number 10 is for gene A. And that's exactly the same for this lady and all of you. But which form of that gene is sitting on that locus is different. So how big is this difference? That is exactly the same gene. It's not another gene. The function is the same, but with a small variation. For example, if gene A responsible for the hair color, I'm just telling this for the simplicity. Obviously, controlling hair color is not as simple as that. But anyway, but assume that gene A, which is sitting in locus number 10, control hair color. So this gene controls hair color in my genome, as well as his genome, and all of you. So basically, the gene A, which is sitting in locus number 10, controls hair color on every chromosome. But you know that the hair colors are different between people. So these different alleles are responsible just for different form of that specific features. So it means that we don't get anybody that on the locus number 10 has gene, in a state of gene A, has another gene called B, which is responsible for height, for example. This won't happen, okay? So this is the terms of the kind of variation that we expect in one gene. So the locus is the same, the gene is the same, the function is the same, but obviously we get some minor changes, which is responsible for these minor changes or minus characteristics, difference in characteristics that we see in humans. All right, but the alleles are different. Okay, so when we look to these two chromosomes, one is paternal, one is maternal, so exactly the same thing applies, what I mentioned. It means that the total structure is the same, but the alleles, which is sitting exactly the same loci for the same chromosome, is a bit different. And this difference is in the content of the nucleotide. For example, in one specific nucleotide, one gene has A, another allele may have C. And this A to C changes create some changes in the function, which is responsible for these minor changes that you see in that specific features. So what happened? Then, if the exchange happened during the tetratransformation, it means that these chromosomes change. So basically, after meiosis, the chromosomes that you have in the gamete is not exactly the same as this one. Instead of having a chromosome like this, then you have a chromosome that some part is from this chromosome and some part of it come from this chromosome. Am I right? Yeah. Okay. So even you cannot say that after meiosis, the gamete in terms of chromosome 1 has two forms. Because on the basis of what your friend said, we assume that after meiosis, the gamete that creates me or creates any of you has two options for chromosome 1, has this chromosome or this chromosome. But in fact, this is not the case. Because you know that after meiosis, the chromosome 1, which is present in the gamete, is not this one, and is not this one.